Good afternoon. This is a great opportunity to present this webinar, which puts together our data on research which our team has conducted. The virus which is the focus of this webinar is EBV, or Epstein-Barr virus, which is also known as human herpes virus 4. Interestingly, it was the work of an Irish surgeon, Dennis Burkett, when he was working in Uganda about 60 years ago, which led to the discovery of EBV as the causative agent of Burkitt's lymphoma. So I'm based at the National Virus Reference Lab, which is on the campus of UCD, University College Dublin. The NVRL is the largest virology lab in Ireland and is a centre of excellence where more than 1 million samples of 1 million tests are performed on more than 370,000 patient samples annually. So the team in this, for the work presented in this webinar, involves a collaboration between the nephrology unit in Children's Health Ireland at Temple Street Hospital and includes consultant nephrologists, professors Atifa Iwan, Michael Reardon, Clodagh Sweeney, Maria Stack and Niamh Dolan. So our collaboration of research projects focuses on the understanding and discovery of biomarkers in the field of transplantation, with particular emphasis on the responses to viral infection in paediatric renal transplant recipients, with the overall aim of improving outcomes in these patients. This is my declaration slide. While the introduction of more potent immunosuppressive therapy has successfully reduced the incidence of acute rejection and improved transplant outcomes, it has also resulted in the high incidence of viral complications. The goal of viral surveillance is the detection of subclinical viral infection that triggers an intervention, one that either prevents progression to viral disease or leads to early diagnosis of viral disease, which is associated with improved outcomes. So with regards to EBV DNA monitoring is currently the main strategy to identify renal transplant recipients potentially at risk of EBV complications. EBV viral replication remains a significant problem after solid organ transplantation and is mostly seen in the donor seropositive, recipient seronegative transplant patients. The detection of high levels of EBV DNA post-transplant or significant changes in EBV DNA are known to predispose these patients to severe complications, which include post-transplant lymphoproliferative disease and possibly death. So we have observed that although clinically well, the patients divide into two cohorts. Cohort 1 are what we call patients who are chronic high viral load carriers, or CHL, who remain asymptomatic despite maintaining high EBV viral loads. The second cohort are called resolvers of EBV infection, or REI, that is, patients who resolve EBV infection. The paediatric transplant recipients included in our studies underwent renal transplantation in CHI at Temple Street from about 2007 onwards. The primary diseases included renal dysplasia, juvenile nephrothesis, posterior urethral valves, cystinosis, and so on. The protocol set up by CHI at Temple Street were for samples to be collected prospectively post-transplant for EBV viral load measurements once a week for the first 3 months after transplant, then fortnightly for the next 3 months, then once a month for the following 6 months, and then at annual review clinics. However, if a patient became symptomatic for EBV infection, irrespective of their follow-up schedule, samples were taken once a week until EBV viral loads became undetectable in 2 consecutive samples. EBV viral loads are determined in whole blood and plasma using quantitative PCR. CHL patients are defined as those who maintain EBV viral loads more than 5,000 copies per mil in whole blood for a minimum period of 6 months post-primary EBV infection or reactivation, whereas the REI patients are classified as those who experience primary infection post-transplant or a reactivation of an existing infection, but who resolved infection and did not have detectable viral load for more than 6 months. The standard immunosuppressive regimen consisted of baxilimab induction, tacrolimus, prednisolone, and mycophenolate. If mycophenolate was not tolerated by the patient, azathioprine was administered instead. The target Saccharomyces trough levels in plasma were 12 to 15 nanogram per mil for the first 8 weeks after transplant, 8 to 10 nanograms per mil for the first year, and then less than 6 nanograms per mil after the first year. Mycophenolate or azathioprine was stopped when a patient had detectable EBV DNA viral load and had developed symptoms, or the viral load increased to more than log 10 to the 5 copies per mil. Thereafter, there was marked improvement in the majority of patients. In our studies, which we have published previously, and in agreement with other studies, there were 4 important factors which tend to broadly separate these 2 cohorts. Young age. Chronic high viral load carriers tend to be patients who were EBV seronegative or naive at transplant, and HLA alleles. In our studies, HLA-A02 expression, HLA-A02 allele expression was predominant in the CHL patients at about 80%. In contrast, HLA-B8 allele expression was prevalent in the REI patient cohort at about 70%. With regards to cytotoxic T cells, we observed reduced or none in the CHL cohorts, supporting the inability to clear virally infected cells. The EBV serostatus of our patients, shown in our previous publication here, depicted on the left, shows where 70 to 80% of the patients under the age of 10 were seronegative at transplant. As expected, the figure drops to about 50% in patients over the age of 10. As you can see here. The graph on the right shows the percentage of patients with detectable EBV DNA over time following renal transplantation, which is over 60% up to 1 year post-transplant and up to 40 to 50% in patients up to 5 years post-transplant. Now, I just wanted to give you a brief background on EBV infection and EBV gene expression. So according to epidemiological studies, EBV is estimated to be positive in about 90 to 95% of the world's adult population. The virus is ubiquitous and infects almost all humans during their lifetime, and following an acute phase, persists for the remainder of the life of the individual. It's a lymphotropic herpes virus and the causative agent of infectious mononucleosis. In the diagram on the left, you can see that like other herpes viruses, following a primary infection where it infects epithelial cells, Epstein-Barr virus will enter the B cells and enter a stage and persist in latency in resting B cells. In the latent phase, you can see there is reactivation and further persistent infection and shedding of the virus. This can occur when there is immune stress or weakening of cellular immunity. On the schematic on the right-hand side, I just wanted you to see the gene expression during the different stages of EBV infection, be it viral lytic phase, reactivation phase, or latency phase. So you can see the different genes that are involved. I will talk about miRNAs later in the talk. And what is important is that the different stages of EBV cycle will depend on replication latency and reactivation. And you can see here that consistently what is being achieved by EBV is immune evasion, proliferation, and apoptosis. And I'll talk about these genes in the next slide. So the EBV genome is illustrated here. As you can see, the EBVs or EBV encoded small RNAs, these are non-coding RNAs, are expressed in latently infected cells, and there's 2 EBVs, EBV1 and EBV2. The BARTs or the BAM H1 right with transcript BARTs, you can see there's many BART genes that are expressed as well. And then you have the BHRF region, which again is highly conserved in EBV-related samples and is actually considered to be a good target for EBV vaccine development. And there's genes expressed here as well. There are 6 nucleoproteins, so EBNA1 through to 6. You can see here, EBNA3, there's ABC, and so on. And there are 3 latent membrane proteins, LMP1, 2A, and 2B. The LMPs are involved in malignant transformation and virus replication during EBV infection. LMP1 has a greater degree of polymorphism than many of the other EBV genes, and gene expression and viral genome replication are generally tightly regulated. There are 4 latency types as shown here, which have been described based on the pattern of EBV proteins expressed, and these are shown here. So you can see in healthy individuals who, for example, are infected and have infectious mononucleosis, generally only the EBVs would be detected or expressed. In type 1 latency, Burkitt's lymphoma patients, you also have EBNA1 being expressed as well. And in type 2 and type 3, for example, you have nasopharyngeal carcinoma or you have PTLD, you can see that more of the EBNAs can potentially be expressed. But also now you see the appearance of the LMP genes as well. This slide shows the gene expression patterns in 8 CHL patients, 9 REI patients, and 10 healthy controls that we studied. And these healthy controls were obviously seropositive for EBV and had EBV infection previously. What you can see is that in the healthy controls, you may remember from the previous slide showing the latency, we could only detect EBNA1. In the REI patients, both EBNA1 and some of the BARTs were expressed as well. However, in the CHL patients, you can see BZLF, which is a lytic gene expressed, as well as LMP1, LMP2, and EBNA1 as well. The expression of the BARTs and the EBVs were highest in the CHL group. And you can see that this really looks very similar to the type 2 or 3 latency as shown in the previous slide. So our results would suggest that these are the patients that should be closely monitored. Now, one of these CHL patients who was HLA-A02 had HLA-A02 allele. We followed her over a period of 3 years. And that's shown here. You can see the months post-transplant and the EBV viral load log 10 copies. So really a log 10 of 6 is going to be 1,000,000 copies per mil. And what you can see is that there is consistent expression of the EBVs and BART genes, and also we saw consistent expression of LMP2 shown here. Interestingly, with EBNA1, there was the appearance of EBNA1, but that then declined. And EBNA2, we were not able to detect EBNA2 over this timescale. What you can see is that when the LMP1 gene, when there's a decline in that, there's an increase in the lytic gene BZLF, and that coincides with an increase in viral load, as you would expect. This patient subsequently, a month or 2 later, developed PTLD, so it can be seen that this type of information may be valuable in identifying patients who may be at risk, increased risk of developing PTLD, and therefore selective reduction of immunosuppression is needed as a mechanism to control EBV replication. Now, on this schematic, I just wanted to show you the function of LMP1, which is very clearly shown here. You can see that LMP1 proteins, the signaling domain here, CTAR1 and 2, recruits the TRAFO-TNF receptor-associated factors, and as a result, results in activation of NF-kappa B and AP1, which will result in cell survival and cell proliferation. On the other hand, you can see activation of BCL2A1, which is anti-apoptotic. And again, you can see these cell functions heavily influenced by EBV genes. So my first call to action slide is this. From our studies, our findings indicate that monitoring of EBV viral load should include monitoring of lytic and latent genes, EBV genes, in patients at high risk of developing PTLD. Changes in gene expression may inform clinical decisions regarding immunosuppression. The target of current assays are unknown. In the NVR, the ARTIS EBV assay is used. The specific target for the assay is not given by the manufacturer. However, the PCR amplification targets a 97 kilobase paired region. As assays can vary, consistent sample type with the same assay is a must. However, there can be unforeseen complications like the discontinuation of an assay. So this is where digital PCR offers a great solution going forward. I want to now talk about our recent studies on EBV microRNA as potential biomarkers in our patient cohorts. So microRNAs are a subset of small 18 to 25 nucleotides of non-coding RNAs, which have been shown to be very stable in blood. They've been demonstrated to play key roles in growth and development, regulation of immunity, cell differentiation, metabolism, apoptosis, cell proliferation, and malignant transformation, and their function has been linked to certain human diseases. So EBV was the first human virus known to encode microRNAs, and the EBV microRNAs are clustered in two regions of the genome, the BHRF1 and the BART regions, to produce 44 mature microRNAs. It has been suggested that EBV microRNAs can potentially manipulate the genome expression in infected as well as uninfected cells due to their ability to be secreted from infected cells in extracellular vesicles such as exosomes. Now, EBV microRNA profiles have been studied in EBV-associated malignancies and in primary infections. However, there are few studies of their profiles in transplant recipients and following immunosuppressive treatment. So I'm now going to describe our findings in plasma EBV microRNA in pediatric renal transplant patients. What I want to show you on this slide is that really these EBV microRNAs can have various functions. You can see here a range of them can block apoptosis, inhibit viral peptide presentation to T cytotoxic cells, initiate cellular replication. But the one I want to draw your attention to is this one, microRNA BART2-5P, which blocks MICB gene expression. And MICB is a MHC class 1 polypeptide related protein, which serves as a ligand for NK cell receptor. So by downregulating expression of MICB, the cell then is not identified to NK cells for cell destruction. Okay, so our patient cohort will consist of 10 CHL, 12 REI, and 8 infectious mono patients. And we looked at 42 EBV microRNAs with 95% coverage of the genome. 4 human microRNAs were determined as well, as these were used to normalize the remaining data. As these samples were collected over a period of several years, a sample quality control using a spiking was performed, as it is important for any qPCR experiment to ensure that the quality of the input RNA is sufficiently high for effective amplification. This study represents the first comprehensive clinical investigation of plasma EBV microRNA expression as potential biomarkers for pediatric renal transplant recipients. The heat map here shows the relative expression levels of the different EBV microRNAs, and in this case, green shows highest expression and red lowest expression. I'd like you to draw your attention in all 3 panels to the BART2-5P, which I've just spoken about. And you can see most of the patients show expression, be it high or low, of this particular EBV microRNA in all of the cohorts. Okay. This heat map shows the relative expression of the EBV microRNAs at various time points in the renal transplant patient who developed PTLD, which I mentioned previously. The pink line here shows when PTLD was developed. So analysis was performed of 10 samples over a 30-month period, and in this patient showed an average of a 24-fold increase, higher levels, 24-fold higher change in plasma EBV microRNA for the BART2-5P compared to the groups you had seen, the CHL, and over 110-fold greater than the REI group. You can see in this patient at all these time points studied, there was high expression of BART2-5P. So just to summarize this part of the study, although there was considerable variation, our results suggest that the BART2-5P, which targets the stress-induced immune ligand MICB to escape recognition and elimination by NK cells, may have a role in sustaining the high EBV viral loads in the CHL cohort. Our findings highlight the important functional roles of the EBV microRNA during the viral life cycle, including immune evasion, cell survival, and proliferation, as well as control of the latent lytic switch. However, future investigation in larger cohort of patients is required to provide additional data. I'm now going to move on to another study which we published recently on the longitudinal analysis of the impact of rituximab treatment on circulating EBV microRNA in 3 pediatric kidney transplant patients. And we did this to improve our understanding of EBV microRNA in the context of what the impact of rituximab on circulating B cells is, what the impact of rituximab treatment on EBV DNA levels is, and what the impact of rituximab on plasma EBV microRNA expression is as well. So these parameters were profiled in 3 patients who received rituximab post-transplant for persistently high EBV viral load longitudinally over a 27-month period. So prior to rituximab treatment, patients received paracetamol, chlorpheniramine, and prednisolone to reduce side effects. Rituximab infusions were administered intravenously. Patients 1 and 3 received 4 cycles of rituximab, whereas patient 2 received 3 cycles. And over the course of this study period, patients received tacrolimus and prednisolone as well. And mycophenolate or azathioprine was held in patients with symptomatic EBV infection, or if the EBV viral load was persistently more than log 10 to the power 5 in whole blood. So you can see the demographics of the patients here, and you can see that high risk factors are associated with all 3 patients in that they were younger than age 10 and they were all EBV seronegative at transplant. They were receiving maintenance immunosuppression with tacrolimus. Patients 1 and 2 received transplants in 2017 and patient 3 in 2016. Both patient 2 and 3 carried the HLA-A2 allele. Now these slides are a little bit busy, but I'll take you through them. On the left is shown the flow cytometry data, and you can see here with rituximab, which targets CD20 on B cells, there is a reduction in the number of B cells, but 10 months later in this patient, post-rituximab treatment, you can see the B cells starting to repopulate in the circulation. What we also noticed was that there was a reduction in NK cells as well after treatment with rituximab. And this was shown by Engquist et al., who reported a drop in NK cell numbers in patients with follicular lymphoma who were treated with rituximab. Now, you can also see that the CD4, CD8 T cell ratios are inverted in these patients as well. With regards to the EBV microRNA expression, which is shown on the right, and here the highest levels are shown in red, lowest in green, but you can see in patient 1 over 26 months, the numbers of EBV microRNA detected varied. There's quite a large number here where you can see a lot of the microRNAs in red, and the same again just prior to rituximab treatment, which is shown here in the arrows. In this patient over the 26 months, the EBV viral load is shown here, and you can see that they are quite high at times at log 10.6, which, as I mentioned before, is 1 million copies. I just want to draw your attention again to the BART 2-5P, which again was expressed really consistently in this patient over the time of the study. I mean, the expression of it does vary, but it is there. A similar format is shown for patient 2. We only had 2 time points for the flow cytometry, and you can see again there's a reduction in the B cell and NK cell numbers. And again in this patient you can see that there were, times which don't always correspond to high EBV viral loads, but you can see quite a lot of expression of the different EBV microRNAs. And if we again zone into the BART 2-5P here, you can see expression of that in pretty much all of the samples that were tested in this patient. In patient 3, there were fewer samples to analyze, but again, consistently in this patient, you can see that at about month 9, the B cells are starting to repopulate in the circulation. And again, reduction seen in the NK cells as well. And for this patient, again, we had very few samples looking at the EBV microRNAs, but you can see that the BART 2-5P, which is here, again, consistent, be it high or low expression, it is there in these patients. So although the EBV DNA viral loads and the microRNA expression was reduced significantly after treatment with rituximab, the reason for the continued detection of the viral loads may be that EBV is known to infect T and NK cells as well, so they could be contributing to the EBV DNA and microRNA expression in the circulation. Second possibility is that free EBV DNA in the plasma could be derived from dying infected cells. And thirdly, rituximab is known to coat the B cells, and this could induce complement-dependent cellular cytotoxicity, resulting in death of the cells and release of the virus and the microRNA into the plasma. So our findings, our small study of only 3 patients, highlight that rituximab treatment may only be effective for patients with only B cells infected with EBV. Larger studies are needed to support our findings, and to call to action, slide 2, biomarkers to accurately assess the degree of immunosuppression in renal transplant recipients to prevent such post-transplant immune-related adverse events are needed. There's an urgent unmet need for non-invasive, personalized precision diagnostics that enable effective monitoring of immunosuppression in organ transplant recipients. The gaps left by histological assessment and late emerging biomarkers of allograft injury provide an ideal opportunity for innovative thinking to extend graft survival and improve patient quality of life. These innovations should potentially allow for customized and cutting-edge immunosuppressive dosing based on an individual's unique immune function and response to therapies, and early identification of under or over immunosuppression that could lead to rejection or infection. Currently, TORC10 or TTV, which is a ubiquitous virus and which has not been linked with any specific human illness, is thought to reflect the immune function of the host. Large studies currently undergoing in Europe involving several centres are looking at TTV, and the group are called TTV Guide. We are also studying TTV levels in our cohorts. However, in my opinion, multiple biomarkers are needed, bearing in mind that both viral and immune factors should be considered together. I'd like to thank the patients and their families for participation in these studies, and Temple Street Foundation for funding to perform these studies. And thank you for your attention.